with this. All right. What we're going to try to be covering today, a little bit more about reactions and some things about enzymes. Then hopefully we're going to get into the macronutrients and then maybe start talking about ATP and how that's actually created. So if anybody recalls, this is where we left off, like endergonic, exergonic reactions. Other terms for these are anabolic or endothermic, or catabolic or exothermic, meaning that like one requires energy to be added, meaning that we're taking a simple molecule, adding energy to it, making it subtly more complex, kind of fighting against entropy, or the exergonic reaction or catabolic reaction, typically taking a more complex molecule, breaking it down in order to normally get energy out of it. And here there's this coupled reactions concept. So typically these are always going back and forth. And that's, where is that? That's illustrated here, more or less. Now I'm going to go back and look at those other slides. But the coupled reactions idea is almost any time that there's an endergonic reaction, we have to feed that by breaking down some other molecules in an exergonic reaction in order to essentially like take energy from one molecule and then add it to another molecule. So one thing I always think about, it's like robbing Peter to pay Paul. If anybody's ever heard that like idiom before, that, or like it takes money to make money. Anyone good with that one too? All right, good. Going back just a little bit. So here, a couple of things on this slide that I want you to like really be aware of. Probably the first thing, does everybody know the chemical formula for glucose? DJ, what is it? C6H12O6. So write that down and know that. That's very testable material, very like quizzable material. C6H12O6, and that's in the top like left-hand corner right here. So most of bioenergetics in regard to exercise is actually regarding the breakdown of glucose in particular. So it is really important for you to know what the chemical formula of it is. So in a biological system, we tend to slowly liberate energy from a given molecule so that we can get the most energy out of it in the most efficient way possible. Now, what we could do is we could just take a glucose molecule and set it on fire and then get a massive amount of energy from it all at once. Now, the problem with that is it's like drinking from a water hose, or like drinking from, like what's the type of hose that like, man, what is it? What is it? Like a fire hydrant? Like what do firefighters use? Like, you know the big hoses right there? All right, this analogy is crappy, so I'm going to move beyond it. So my bad, right? It's like, so that's essentially not super effective. So we have an orchestration of like numerous enzymatic processes in order to get the most energy out of a molecule as possible, kind of illustrated here. Something else that I really want you to understand is up here in this top left-hand corner and the bottom right-hand corner. So never lose sight of this is mostly what we're doing. So we're taking a certain macronutrient, in this case glucose. We breathe with it, so 6 O2s. I might just know that, C6H12O6 plus 6 O2s. Might need to know that. We breathe, and then on the tail end of it, we get 6 carbon dioxides and 6 waters. And along that way, we're taking chemical energy from the glucose. That chemical energy is being transformed, this whole bioenergetic idea, into mechanical energy, and in our case, muscle contractions. That's really what we're the most concerned about. Everyone good with that so far? So C6H12O6 plus 6O2 yields energy, right? And 6 CO2s and 6 waters. So one of the big questions is like if anybody had like Culver's last night, or like, I don't know, pizza, or like toppers, anything like that, where does it go? Anybody? So last night I ate my dog's dog food because it's great for humans, and whenever I breathe out right there, that's my dog's dog food coming out. Right? So like keep that in mind whenever we're going forward. All right, everyone good with this? Okay, next thing here. A couple of things that are testable, a couple of things that aren't super testable. y'all aren't really in a humorous mood today. That's all right. It's okay. Moving on. Endergonic reactions versus exergonic reactions. I want you to notice where the directionality of the reactants to the products is going in both of these. So in terms of the amount of energy within a substance, an exergonic reaction has more in the reactants, then it is broken down, and then we have less energy in the products. Everyone good with this? We'll have examples coming up. Next thing, the endergonic reaction. Reactants, they have less energy in them. Then we add energy through some mechanism, typically enzymatic mechanisms, and then the products end up being higher in energy. Everyone good with this? Right? Okay, now here's a thing that was never explained to me whenever I was in your shoes, and if you go to grad school, I think it's important for you to know. And if you don't go to grad school, I think it's also important to know. But Gibbs free energy. Hopefully you've read this term in like different like textbooks, hopefully our textbook in particular, but likely in like other biology classes or what have you. Does anybody know what Gibbs free energy even is? No? Well, like whenever I was in your shoes, it was just a memorizable point. That like I was like, okay, Gibbs free energy, and this one, like delta G, like less than O, right there. That's an exergonic reaction. I just memorized that, that's all it was. But here's what Gibbs free energy is. It's a thermodynamic quantity equal to the, now I do have a bit of a speech impediment, so words with L's are somewhat hard for me. Enthalpy, I believe is how it's said, but I typically will say enthalpy because I don't know how to speak, of a system or process minus the product of entropy, which is the second law of thermodynamics, and the absolute temperature. So here's an equation I want you to know. That that quantity of energy within like a macromolecule or within a totality of a chemical reaction, it's equal to this H, which that H stands for enthalpy, which is a measurement of energy in our thermodynamic system. Now, that's enough for us right here. That's enough for us right there. What constitutes H? It's U plus PV. So U, that's the internal energy, and then P, that's the amount of pressure, and V is the volume, right? So amount of energy, and if we have a higher pressure, like less volume, then that increases the enthalpy. Now, if we have a like larger graduated cylinder, say, that is somewhat like inversely proportional to the amount of pressure, and that would end up decreasing the enthalpy. Now, I'm not going to test you about any of this stuff like super in depth, other than more or less just the symbology right here. Everyone good with that? Yes, Courtney? Do you need to know what each of them mean? Yes. 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 Okay, so that's the enthalpy. Now let's look at the temperature and the entropy side of the equation. So typically, things tend to go downhill towards disorder. Hopefully everyone knows this. So the classic example of explaining entropy, which is notated here by the S, is that after you clean your room, it tends to get unclean without you doing anything, right? Has everyone heard of this, right? Wait. Alyssa, you look like you'd never heard that before. Oh, you have? Okay, good. I was about to say, like, what are they teaching over there? That's ridiculous. So things tend to move towards disorder. That's just kind of the chaotic nature of the universe. And with temperature, so that overall makes things somewhat less efficient. So we lose energy the higher temperature and the more entropy are happening. Does that make sense to everybody just by looking at this equation? So G standing for free energy, meaning energy that we can use to do something. If we heat up that reaction, meaning increasing the T, and increasing the T has somewhat of an impact on entropy, that takes away from the H, which the H is the amount of energy. And you can also decrease the amount of energy in a system just with the H by, say, I don't know, manipulating the pressure and the volume. Is everyone good with this so far? Right? It's a bit esoteric, but just diving deeper into how these reactions work, that's some stuff I wish someone would have told me whenever I was in your shoes. So moving on from there. Already talked about coupled reactions, so going to move on from that. All right, oxidation and reduction reactions. We're going to be fairly concerned with these, particularly because we have something in the mitochondria called the electron transport chain. So oxidation or reduction is really important because that is removing an electron or adding an electron. So there's an old acronym that helps people remember this. Does anybody know this acronym? What is it? Oil rig. Of course it is. So oxidation is losing, meaning removing an electron. Reduction is gaining, meaning gaining an electron. Now, it's somewhat counterintuitive because typically most of us think, whenever we reduce something, we're going to have less of it. But the idea here is that it's actually reduced in charge. So it's adding an electron, which is negatively charged, to the whole molecule, thereby reducing it. Is everyone good with that? Okay. So thus far, oxidation, reduction, that's all I want you to know for now. Now, within glycolysis and the Krebs cycle, we have two really important electron carrier molecules, or electron transporters. And I want you to know what these are. Nicotinamide adenine dinucleotide and flavin adenine dinucleotide. So we need to know what these are. Now, you're never going to have to spell that on the test because that would be absurd. This isn't an English class. Even though those probably aren't English words, probably Latin, I think. We need to know what the oxidized and reduced form for NAD and FAD are. Now, going back to genetics a little bit, you can actually measure within a muscle cell the concentration of NAD relative to NADH. And if the concentration tips one particular way, meaning a lot of NAD relative to NADH, that can actually stimulate mitochondrial biogenesis. Meaning that we're somewhat at an energy deficit within a cell. And hopefully that will make sense as we start going through glycolysis. But where it stands right now, the oxidized form, NAD+, right? Everyone good with this? Oil, right? Oxidation is losing, so NAD. Has less, reduced, gaining, meaning that it just gained a letter. Is everyone good with that so far, right? So far, so good? All right. Now, let's look at FAD really quick. So oxidized FAD, then reduced FAD H2. Now, does anybody drink energy drinks? Right, like you all should, especially how you look right now. So energy drinks frequently have 100, 200, 300, some odd percentage of your daily requirement of different B vitamins. So right here, niacin and riboflavin. Those are two B vitamins that the idea is they constitute that whole electron carrier molecule. So if you consume more, then what happens? If we consume more of these water-soluble B vitamins. What's up, Hannah? Nothing? Oh, okay, right. Ultimately, that's actually what happens, right? So Hannah was hop, skip, and a jump before me, what I'm trying to say right there. So the idea is how these energy companies, or how these Bang energy drink companies have these different things. First, they put a lot of caffeine in it, which is actually really effective. But these increased B vitamins, the idea is that if you consume more of these B vitamins, that just law of mass action, we're going to get more of these electron carriers into the cell. Right? So if you just continually feed it, then we'll have so much of it that we're going to be able to do the electron transport chain a lot better. Does that make sense to everybody Now, like how it actually seems to work is a negative feedback loop. You consume too much of these and you just urinate them out functionally. And it might actually reduce the content of like these carriers within the cells, but like more research needs to be done on that. So there, niacin, riboflavin, that's at least the idea behind why those companies are doing that. Like here's another picture like out of the book that could be interesting if you're really into like molecular structures. I'm not going to make you draw any of this, of course, but it is fairly cool. All right, moving on to enzymes. Any questions about those electron carriers? Functionally for the test, I just want you to regurgitate what I said for what's going on with those. All right, so enzymes, these are catalysts that speed or regulate the rate of reactions, more or less. So overall, they're lowering the energy for activation, lowering the activation energy, which I'm going to show you in a picture, like what's happening with that. Now, there's a couple of factors that seem to regulate how fast enzymes work. So the first one being temperature. So within a particular range, if we increase the temperature of a cell, enzymatic reactions go a little bit faster. Now, there is an optimal range with this. That's overall the main reason why we warm up before we exercise, which I feel is a bit dubious, but like I'll explain why in some time to come. Now, pH, that can also alter the rate of different reactions. So that one is even tighter than with the temperature. The next thing being substrate concentration. So I think the easiest thing to understand with substrate concentration is if you consume more of a given substrate, and in this case, I want you to think carbohydrates. So if you consume more carbohydrates, then that's going to increase your body's ability to burn carbohydrates because you're going to have an excess of them. Does that make sense to everybody so far? So that's consuming more carbohydrates leads to more activity in the carbohydrate oxidizing pathways. Next thing is inhibitors and activators. So the primary activator type that we've been talking about is the phosphorylation cascade. So if we put a phosphate group on a given enzyme, then it might change the shape of it just enough to where it actually increases its activity. Everyone good with that? Good? Okay. Like a couple of things. There's two different models that I want you to be aware of. So the lock and key model and then the induced fit model for how enzymes work. So here, let's get through this slide first. Here's the idea. We have some reactants and then like a certain amount of activation energy, and this is overall what enzymes are doing. So due to entropy mostly, almost any chemical reaction that's like going downhill in terms of being an exergonic reaction will occur at some point in the absence of enzymes. But an enzyme essentially reduces the amount of energy needed to get that ball rolling. So if that didn't make sense to you before, hopefully these pictures illustrate that. Here, I'm going to skip over that slide. That doesn't seem to matter so much. All right, here, the lock and key. Lock and key. Whenever we have an enzymatic reaction, there's a couple of parts that I want you to be aware of, right? So at the beginning of it, we have reactants, then we have products at the tail end of it. So essentially, we have an enzyme and either one substrate or two substrates. Those substrates fit into the enzyme like a lock and key, meaning that there's effectively like one enzyme that was more or less put together to fit those particular substrates. Now, that's how it was taught for a fairly long period of time, and I think there's still some evidence to believe this. Whenever those substrates go into the enzyme, which keep in mind this enzyme, that's just a protein, so if we had a different like missense or nonsense mutation, we could completely change the shape of that, right? Is everyone good with that? Right, so kind of tying back in the genetic stuff that we talked about before. So adding the substrates into the enzyme, then we get something called an enzyme substrate complex. And then at the tail end, we have the enzyme just kind of floating off. It did its job. It's ready to be used again. Now we have different products. Hopefully everyone's good with that so far, right? Good? Okay, moving on. Now that's the lock and key model, meaning that this enzyme was essentially structured to deal with particular substrates. Something else is the induced fit model, meaning that really the substrate and the enzyme, or I guess more so the enzyme, has a particular shape that can fit numerous different things, and through some type of modulation, the substrate will be stuffed into there, and there's a conformational or some type of shape change to both the substrate and the enzyme. Now this makes a little bit more sense to me, particularly for something that you don't have. So I would write this down. Allosteric enzymes. So here's the idea here. That the big purple thing, that's the enzyme. The substrate right here, that's the little blue pill. So I guess the modulator is the little red pill. Matrix? No? I thought of that just now. So not as funny. Cool. Moving on. So essentially what's happening here, and this makes a lot of sense to me, is that we have some type of inhibitor or activator that bonds to this modulatory subunit within the enzyme, which will alter the shape of the whole enzyme that either increases or decreases its ability to work with a substrate. Everyone good with this? Right? So, let's just take this for example. If that enzyme is a given charge, and we put a phosphate group on it, it's going to alter the opening right here, which is called the catalytic subunit. That's going to change the shape to where it will either more readily work with something or more readily not work with something. Hopefully that makes sense to all of us. That we can put something into one side of the enzyme that's going to overall change its functionality. Right? Is everyone good with this? Everyone good with this? One thing, for example, this is how cholesterol medicine works. So statins, it's what is it? It's an HMG-CoA synthase inhibitor. So it stops this one enzyme from working, and then you don't make cholesterol anymore, in your liver. So that's kind of a cool thing going on there. All right. Hopefully you understand that that whole allosteric and induced fit model. All right. Something cool. Now, this is a little bit more medical than actually exercise related, but we can figure out if a particular cell, ruptured or is broken or isn't working, based upon if something leaks out into the blood. So think about it like this. Say you have a heart attack, right? If you have a heart attack, normally that means that somewhere in the cell membrane, there's going to be death and a breakage. And then enzymes that are inside of the cell are going to leak out into your bloodstream. And then we could take blood from you and see if a given enzyme or a protein molecule is circulating in your blood. And that's going to tell us if you had a heart attack or not. Everyone good with this? Right? Now, there's all sorts of different types or enzymes that we can measure, ALT, what was it? Alanine aminotransferase. If you have a high amount of that in your blood, that typically means that you have some type of liver damage. But here are the ones that I want you to know mostly. Lactate dehydrogenase and creatine kinase. So if we have a high amount of lactate dehydrogenase in the blood, that's normally related to a heart attack. Same thing for creatine kinase. Now, I'm going to be talking about those two enzymes quite a bit going forward. So something I want you to know is that lactate dehydrogenase has 4 subunits to it. Creatine kinase has 2 subunits to it. Now, lactate dehydrogenase, those 4 subunits, they can be a different mixture of 2 different flavors. So there's a heart-specific form of lactate dehydrogenase and a muscle-specific form of lactate dehydrogenase. So whenever we're measuring it in your blood, we would measure for the heart-specific one. And one cool thing with endurance exercise adaptation is whenever you start doing a lot of cardio and things, if you have a lot of the muscle form of lactate dehydrogenase inside of your muscle cell, it's actually going to start shifting you a little bit to have more of the heart-specific one. Is that interesting to anybody? Like, have you ever felt like a burn in your heart, like not acid reflux, right? So shifting in that direction is kind of cool, at least to me it is, that like on that, like has anyone ever thought like whenever you're doing cardio that like I'm actually making the lactate dehydrogenase enzyme in my quad less like a muscle lactate dehydrogenase enzyme and more like a heart dehydrogenase enzyme? No? It's pretty metal. Right? I think it's cool. Okay, cool. Creatine kinase has 2 different forms of it. It has a muscle and a brain version, which, to my knowledge, we actually don't shift into the brain version from the muscle version from doing exercise, because that really has less to do with, at least the brain version has less to do with exercise adaptation. Okay, good. Moving on from there. Classifications of enzymes. I want you to know these 3, but really the kinases and the dehydrogenases are probably the most important ones for us to know going forward. So a kinase enzyme, bless you. This adds a phosphate group to a given molecule. A dehydrogenase enzyme, this removes a hydrogen. And we've got oxidases and isomerases and things like that. Those are less important for the totality of bioenergetics. But kinases and dehydrogenases, whenever we're going through these pathways, those are like all we use. So be aware of that. All right. So, gosh. Oh, man. All right. Talking about this a little bit. Already talked a little bit about the temperature increases. Something that I want you to know here with the temperature increases is called the Q10 effect. So big Q, 10, Q10 effect. So go and write this down. Q10, temperature coefficient, is a measure of the rate of change of biological or chemical system as a consequence of increasing the temperature by 10 degrees Celsius. So I want you to pay attention to that 10 degrees Celsius. So here, a moment ago, I talked about maybe the dubious nature of warm-ups. Yes, Nick? Yes. Sorry, Nick. All right, are we good on this one? Maybe? Maybe not? Okay, cool. Good here? All right. So recall how I said temperature, increases in temperature, increases enzymatic activity a little bit. So here, think about enzymatic activity as our ability to like break down carbohydrates faster. That would be a good thing to do whenever we're like running in a marathon or running in a 5K or we're lifting weights or anything like that. I talked about the Q10 effect because that's essentially the level at which the enzymatic activity is really going up or not. So here's a number that I think you should just solidify in your head. So 37 degrees Celsius, that's the general core temperature for most individuals. That is 98.6 in American Freedom Units. Hopefully you know that. So if we're talking increase Q10 effect, so like follow me on this. If we went from 37 degrees Celsius to 47 degrees Celsius, what do you think happens? Yeah, you die. You die. So like whenever we're talking about like the Q10 effect, like bear this in mind. Like the whole idea of like you need to get your muscles warm in order to like get increased enzymatic activity, it's not really that much because we cannot increase our core temperature by 10 degrees Celsius. Now, here's a hole in what I'm saying. You can actually increase your muscles by 10 degrees Celsius. So, like I was doing a little bit of math earlier, right? And say your typical skin temperature is around 33 degrees Celsius. Like you could get your muscles down to like 30 degrees Celsius or so, which is like 86 degrees Fahrenheit. And then we could increase that up to 40 degrees Celsius, which is around, what did I write down? 104 degrees Celsius, 104 degrees Fahrenheit, I'm sorry. So that could actually happen. So overall, the idea with this increase in like temperature, we have a little bit more wiggle room with our muscles, but like our core temperature, if it really gets beyond like 42 or 43 degrees Celsius, then we start having heat strokes. So really, it seems to me that warming up is mostly a ritualistic thing. So if anybody is cool with that, because you see here, this enzymatic activity, it just goes a hair up whenever we go from 37 to 40. So hopefully everyone's good with that. All right. Here with the pH, it's effectively a fairly similar range, like around 7.4 or so, 7.6, something like that, for within our muscles. So like some modulation within there can actually increase enzymatic activity. So yeah, I'm not going to talk about that yet. All right. Any questions about enzymes? No? I'm sorry it's a rough one today. But hey, it's the tail end of things that we're going to be tested about for this test. All right. So now let's get into macronutrients just a little bit. And we'll get through as much macronutrients as we possibly can, and then we'll call it a day. Cool? All right. Cool. So carbohydrates. Couple of terms. Monosaccharide, disaccharide, polysaccharides. So mono means one, effectively. Disaccharides, that means two. Polysaccharides means many, right? So what are the monosaccharides? Does anybody know? Glucose is one of them. That's the main one that we use. That's the blood sugar one. Anybody know? Fructose, that's another one, fruit sugar. What's up, Osa? Sucrose, that's actually a disaccharide. So sucrose would be the combination of glucose and fructose, table sugar, effectively, right? So the next one, a lot of times people don't think of it very much. Galactose. Galactose, it sounds like a Star Trek sugar, right? So glucose, fructose, and galactose. Those are the three main monosaccharides, or they're the only monosaccharides, actually. Now, for fuel, something that I want you to know, any cell in the body can use glucose. Everyone good with this, right? Like your brain can use it, your muscles can use it, your liver can use it, whatever. Now, the other two, fructose and galactose, only your liver can process it. So whenever we're consuming table sugar, as Osa was talking about, sucrose, one of the disaccharides, that fructose can only be processed in your liver. Right? Everyone good with this? Right? So what good is fructose for muscle contractions? It's really not. It's really not. Now, there is some research that indicates if you consume fructose, your liver will use that, and that's going to free up more glucose for your muscles to use. That's actually why Gatorade is made in the particular fashion that it is. So it's not like fructose is a terrible thing, but it's not going to go to your muscles. But that's about all I wanted you to know, at least in regard to mono and disaccharides. Now, a few more terms: glycogen and glycogenolysis and glycolysis. I want us to know what these mean. So glycogen, 2 places it's stored that are relevant to us. It's the storage form of glucose in the liver and the muscle. Now, whenever we get into exercise metabolism, we're going to be talking about burning muscle glycogen and blood glucose. So burning muscle glycogen, that is the storage form of glucose within the muscle. Blood glucose, we derive that from liver glycogen or from your diet. Everyone good with this so far? Right? And we'll do some calculations coming on. All right, next thing, glycogenolysis. Many people use this term interchangeably with glycolysis, and it's incorrect. Glycogenolysis is the breakdown of glycogen to glucose. So this is going to be really relevant for whenever we start doing higher intensity exercise and we have to use muscle glycogen in order to fuel it. So you would do glycogenolysis and then glycolysis whenever we're doing high intensity exercise. Okay. Hopefully, that makes sense. Here, just another bit of a term for glycogen, and I'm going to show you a picture that I think is fairly cool. Glycogen, it's really just numerous branches of glucose molecules with something at the core of it called glycogenin. And one of my oldest friends, his name's Cody Hahn, in a biochemistry class, he said this. And I don't think anyone in here is going to think it's deep, but I'm okay with that. He said to our biochemistry teacher, so you're telling me at the core of stored glucose is glycogenin, which is a protein. So if anybody remembers protein, of the utmost importance, kind of the deepest thing, right? So what glycogen is actually stored to, or bound to, is a protein. Is that interesting to anybody? Right? No? All right. It's, well, Cody was cool. All right. So glycogen structure. This is effectively how it looks. Essentially, here's the glycogenin here at the center point of it, and then branching out from it, these are numerous glucose residues. So we could get in a little bit more like an alpha-1,6 residue, but that doesn't really matter too much. What I want you to understand is that this is stored within your muscle and liver, and it looks somewhat like this. And then enzymes get around it to break down glucose molecules from the end of it working to the center. Interesting to anybody?
So this is just the most efficient way to store it?
Yeah. Yeah. Yes.
Most of it, smallest amount of space?
Yeah. Really in just some type of I don't know, sphere more or less. So, yeah. All right, good with that, everybody? All right, here, 5 minutes or so, I think I can finish talking about fats. So first off here, everything listed right here is a lipid, which essentially just means it's not dissolvable in water. But there's a couple of primary aspects that I want you to know. So fatty acids. This is fat utilized by our muscles in different cells for energy. We're going to convert that to ATP. Now, triglyceride. This is the storage form. So we store fat in our fat cells, obviously, called adipose tissue. And we also store it within our muscles, called intramuscular triglycerides. So I want you to write that down. We store a fair bit of fat in our muscles, IMTG, intramuscular triglycerides. That's normally the acronym that's used for them. So as we were talking about before, and this maps on really well, I think, with glucose, or carbohydrates, we store it in the muscle, and we also have a blood form. So we have muscular, like glycogen, right? Carbohydrates stored in the muscle. And then we have blood glucose, which is essentially derived from liver glycogen. We have a fairly similar thing with fats. We store some in the muscle, intramuscular triglycerides, and then we will have circulating fatty acids, which come from adipose tissue or fat cells. So essentially both of those are the main forms of fuel that we use to exercise. So we have fats and carbohydrates, and both are stored in two different places. Everyone good with this? It's almost like it was designed. I think it's pretty cool, right? And here, I'm not really going to talk about phospholipids or steroids. They are fairly interesting, but moving on from there. A couple of points with the lipid structure. We have a multitude of different types of fatty acids, and I want you to pay attention to the ratio of carbon to hydrogen to oxygen with this. And this is going to be really important for lab tomorrow. So a triglyceride, this is a glycerol backbone, which is only made out of carbon and hydrogen. Everyone good with this? Right? Okay. I'll come back to that in a little bit. Then we have fatty acids stuck onto it. So these fatty acids are mostly carbon and hydrogen and a little bit of oxygen. So in order to break these down, we have to breathe a lot more in order to break them down because there's not a lot of oxygen on them. Does that make sense to everybody? So tomorrow, whenever we're talking about the respiratory exchange ratio, that's effectively what's going on here. So we can figure out what we're burning for fuel based upon how much oxygen is going in your face and how much carbon dioxide is coming out of your face. Watch the video for lab tomorrow, and it'll make more sense, especially whenever we're measuring it. But I think this is pretty cool. Now, with these, there's a few different flavors of fatty acids that we actually burn for. What if I just stop here? Are y'all good with that? Of course y'all are. Okay, cool. So here, I'll talk about the types of dietary fats on Wednesday, and that'll be the last thing that I'm going to test you on. All right? Cool. See you in lab tomorrow.